Center for Biological Research in Madrid. Afterwards, she was a postdoc at the Spanish National Center for Biotechnology at Madrid and then joined the Plant Research Laboratory at Michigan State in 2018. Her main research focus is on the effect of the addition of heterologic zincs to cyanobacterial cells on the photosynthetic efficiency, as well as their capacity to act as photoprotective mechanisms. So I'm looking forward very much to this talk and see you all at the Q&A session.
As he mentioned, I am Maria Santos. I work in Plant Research Laboratory in Michigan State University, and today I'm going to talk about metabolic engineering of cyanobacteria. This is the outline that I'm going to follow in this presentation. I'm going to start defining what is metabolic engineering, why cyanobacteria could be an interesting host for biomolecular production, how to use some genome scale models for engineering cyanobacteria metabolism, what tools has been developed for cyanobacteria. After that, I'm gonna focus in a couple of examples on metabolic engineering, omega-3 fatty acid, limonene, and I would like to finish this presentation to showing some future challenges in metabolic engineering, as well showing some useful resources for your possible research in this topic. What is metabolic engineering? Metabolic engineering can be defined as the modification of metabolic pathways, also the regulatory processes, with the aim of improving the production of metabolites. It could be biochemicals, pharmaceuticals, biofuels, or other biotechnological products. In this metabolic engineering process, we use normally a cycle that is compound of 4 steps. It starts with the step of design, in which we need to define which compound we want to produce. We need to select the host. In this case, we are going to use cyanobacteria, which enzyme pathways are involved in the production of these compounds. We can use synthetic design to introduce these pathways in the host. And after that, when we have decided all these steps, we need strain optimization. With all this information, we jump to the next step, build a step that we build the strain with all the modifications that we want to introduce. In this step, there are new tools that we can use as CRISPR-Cas systems. We can use adaptive laboratory evolution, even synthetic codons or chromosomes. Once we have built our possible strains, we need to test the production of the specific compound. It could be really helpful if we have high-throughput screening methods, and also if we perform multi-omic analysis, we are going to get a lot of information that is going to be useful in the step of learn. In this step, we are going to accumulate all the information that we have obtained in all the steps, and it's going to be useful for the next step of design, build, test, and learn. What are the considerations for more efficient design, build, test, learn cycles? We can ask before starting these cycles different questions that are going to allow us to decrease the number of cycles that we need to do. First one is, how strongly should I express my gene? If there are multiple genes in the pathway, should they be expressed at different levels? Should my genes be on all the time or only induced under a certain set of conditions? How do I optimize the flux of carbon away from the endogenous pathway and through my pathway? How do I balance the intermediate metabolites within my pathway in order to maximize the total flux? And finally, how will my introduced pathway interact with the cell physiology? Toxic to the cell or negatively impact upstream processes of photosynthesis? Presentation we're going to have different response depending on the compound that we want to produce. In this presentation, we are focusing cyanobacteria as hosts. We are going to describe what are the advantages and disadvantages of use these microorganisms for bioproduction. Cyanobacteria offers some advantages compared with heterotrophic organisms as E. coli or yeast. because the media that they use is very simple, and also the carbon source that this media contains is very cheap. Also, if we compare with plants, cyanobacteria show higher efficiency of solar energy capture and conversion, as well as they don't compete with arable land or potable water supplies. Moreover, the processes for production in cyanobacteria are carbon neutral production process, because instead of producing CO2, these organisms are able to consume this molecule. Cyanobacteria are easy for genetic manipulation, comparing, for example, with algae, and the leftover of the biomass production is a valuable product. And also cyanobacteria, there are organisms that produce natural secondary metabolites. As you can see here, cyanobacteria, depending on the species, obviously they produce some specific compounds, sugars, alcohols, terpenes, fatty acids, etc. However, there is also some disadvantages that these organisms they need to overcome. Cyanobacteria has some slow division rates compared with E. coli and yeast. However, more and more new cyanobacteria have been discovered in the last years that they show fast-growing phenotypes. Also, comparing with E. coli and yeast, the limited number of genetic tools available is a step that we need to improve. In most of the cases, we use genetic tools that have been developed for E. coli, and the information that cannot be directly applied, as I will show you later. Also, in order to use these organisms for large-scale production, we need to develop a system that allows us to do that. Cyanobacteria are polyploid organisms, and when we introduce a modification in the chromosome, we need to be sure that all the copies contain this chromosome, and because of that, the segregation processes are longer than normally. And also because we introduce this modification in the chromosome, this fat produces genetic instability of the telomerase pathway. And comparing with other prokaryotic organisms, the number of sequences or cyanobacteria is relatively low. In this graph, I am showing how many number of sequences we have of other prokaryotic organisms, and cyanobacteria, as you can see here, the percentage is very low. In this table, I am describing what are the most common cyanobacterial strains used in metabolic engineering. We have Cyanecocystis, that is a freshwater unicellular cyanobacteria with small genome size, 7 plasmid, and a lot of DNA transfer methods has been developed for these cyanobacteria. In the second place, we have Cyanecococcus elongatus, PCC 7942, that has also a small genome that contains normally 1 plasmid, and in some cases, another extra plasmid that is not essential. This is also a freshwater unicellular that can be transformed for conjugation, natural transformation, or electroporation. Close related species that was discovered recently in 2015, Cyanecococcus elongatus UTEX 2973, is quite similar to 7942. In fact, the genome is same size, only contains 1 plasmid, and the difference between these two species is the little amount of single nucleotide polymorphisms that allow to this strain to grow faster. And one of these SNP is affecting one of the proteins involved in natural transformation. That's why it's only able to be transferred by conjugation. We have also uralin cyanobacteria 7002 that contains 6 plasmids, and small genome size. The main problem with this cyanobacteria is it requires vitamin B12 to grow, and it's not. It's going to be really expensive if we want to use for industrial production. And the last one that we have in this list is Nostoc 7120. It's a filamentous cyanobacteria. It could be interested in the processes that we need to recover the biomass because the product is accumulated inside of the cell. As I mentioned before, more and more cyanobacteria have been recently discovered with fast-growing phenotype. In fact, in 2018, Cyanecococcus elongatus 11801 was isolated from India. This year, close neighbor to this one that I have shown has been presented, and also this year another one, 11901, has been recently described as a cyanobacteria for high biomass production. Even though cyanobacteria offers some characteristics that could be useful for metabolic production, we need to modify these wild-type strains to generate a chassis that allows us to produce the specific compound. In order to choose which strain we want to use, we are going to focus in the strains that with genome sequencing shows high quality annotation. And also, we should choose cyanobacteria which genetic tool has been developed, because for metabolic engineering, we are going to need several parts. I'm going to show in next slides: promoters, ribosome binding sites, terminators, riboswitches, etc. Also could be useful if we choose a cyanobacteria which metabolic models has been developed, because we can use in silico these metabolic models to predict which genes we can overexpress, downregulated, or delete to increase the production of a specific compound. Also, if we have some previous physiological characterization, transcriptomics, and other omics analysis, it's going to be really helpful to figure out what are the conditions to improve the production of a specific compound. Not in cyanobacteria, but in E. coli, reducing genome strains have been developed, and it could be an interesting area that could be explored in the future in cyanobacteria to get cyanobacteria with reduced genome that allows us to increase the genetic manipulation. And finally, if we have some cyanobacteria with mutant collection, for example, knockout library has been developed, could be useful if we want to work with a specific competitive pathways that can be removed. And also, if we have some high throughput screening platforms that can be used for this screening, is going to be speed up the processes to generate our final chassis. With all of this, we are going to focus how we can apply first genome scale models for engineered cyanobacteria. To start this part of the presentation, I would like to define what is a genome scale model. It's a large-scale stoichiometric model that describes metabolic pathways as stoichiometric coefficients and mass balances of participating metabolites, and are simulated using numerical optimization. In other words, this is, for example, the metabolism or our cyanobacteria. We are going to have a matrix, as you can see here, and in the rows, we are going to have the reaction, and in the columns, the metabolites, and these stoichiometric numbers represent the reactions that involve this metabolism. We can use different constraints and algorithms that are going to allow us to identify which gene we can overexpress, downregulate, or knockout. After that, when we identify these modifications in silico, they can be used in vivo to be manipulated the genome of the specific strain and increase the production of the target chemical. One example we have here, we have in our metabolic model, we need to apply some constraints. In a steady state, the flux of all reactions in the cyanobacterial species is going to be equal to 0. We need to establish an lower and upper bound to some specific constraints. Normally, it could be the light intensity that we use in the production, the CO2 percentage that we use. With these constraints, we are going to get an allowable solution space. And the objective or to constrain a little bit more this space of solutions, we can maximize the objective function that in this case is going to be the production of a specific compound, and the algorithm is going to give us the optimal solution to improve this production. A lot of algorithms have been developed to perform this, to generate or obtain this optimal solution, and some of them are specifically involved in getting which genes we can delete, other ones which genes we can overexpress, and in some cases, some algorithms can identify which pathways or reactions we can include in our species to increase the production of a specific compound. At the end of this presentation, I'm going to focus on one example that they use this algorithm of force. In this slide, I'm going to show how cyanobacterial genome scale models have been developed. First, cyanobacterial models were very basic. They included only information about central carbon metabolism and some rudimentary light reactions. But the recent model has been improved more and more because they base the information on genome annotation and literature. They can study information for databases as Cyanobase, Transport Database, and Metaseq. And the genome scale models can be obtained quickly if we use some tools that has been developed for rapid generation of draft. However, these draft, they need to be still curated manually to correct some mistakes that these tools can be produced. And as you will know, cyanobacteria contains a high percentage or unknown genes. And with these tools from gene protein association and gut feeling, the information that can be added to the model can increase their accuracy. And also, models can be validated using essentiality gene information, comparing experimental phenotype with genome scale model prediction. We can estimate how accurate is our model. And in the future perspective, this model can still be improved, obviously. It can be improved using integration of omic datasets, and also improving the modeling of light reactions, alternative electron pathways, and photorespiration. And could be really, really interested if we use the information that has been developed for kinetic models and introduced in these genome scale models. In this list, you can see all the cyanobacterial genome scale models that have been developed during many years. Cyanocystis has a lot of genetic genome scale models and include more reactions and genes and metabolites. The second case with more metabolic models is 7002. These genome scale models I mentioned previously offer some advantages. They can be used in silico to determine which modifications we can perform in our model states to produce a specific compound, chemical compounds. At the end of this presentation, I'm going to explain this example where limonene production was increased using this algorithm. For doing metabolic engineering in cyanobacteria, we need some specific tools. In the first place, we need to use some plasmids to introduce the modifications in the cells. We have different type of plasmids in cyanobacteria we can use. In all the cases, we are going to have an original replication of E. coli that allows, because the modifications or the construction of this plasmid we perform in E. coli. We have first kind, conjugative and self-replicative plasmids that they need an origin of transference, because normally these plasmids are transferred from E. coli to cyanobacteria, and they need an original replication of cyanobacteria, normally are based on some endogenous plasmids that this cyanobacteria contains, or we can use a broad host range original replication plasmid are RSF1010. The second type of plasmids that we have are conjugative and integrative. In this case, we are going to have an original transference for transfer for E. coli to cyanobacteria, and also some neutral site regions upstream and downstream that are going to allow us to integrate our construction in the genome. And in the last case, we have integrated plasmids that are going to be directly integrated in the chromosome, and these plasmids normally are transformed in cyanobacteria by natural transformation. Normally, there are going to be some cyanobacteria that are not natural transformation, and in this case, we are going to need to use conjugative integrative plasmids. Between these two regions, we can introduce different parts. We are going to have always an antibiotic resistant to select our transformant. We can use different promoters, ribosome binding site, riboswitch, and different terminators. All these parts I'm going to explain in the next slides. As in other organisms, we can use modular vectors for engineering cyanobacteria. In fact, many years, BioBrick plasmids that were developed firstly for E. coli and Bacillus subtilis have been used in cyanobacteria. But recently, a specific battery of this BioBrick called Cyanobricks has been developed specifically for 7942, using different neutral sites, antibiotic resistant promoters, and the gene of interest. And as in the case of BioBrick, this Cyanobricks follows the rules of nomenclature for this specific plasmid. In the second place, another system that has been developed for E. coli are modular cloning system that is based on the Golden Gate cloning system, and it's based on the use of different levels at 0. We have promoter, terminators that we are going to join in the level 1, and after that we can join into the plasmid. A similar system has been developed recently for cyanobacteria called Cyanogate. And in the last example, we have cyanobacter. This cyanobacter has the different parts cloning in different plasmids or vectors with that are gonna contain overlapping GC adapters upstream and downstream of our part. Once we have selected our the different parts that we want to clone, we are gonna restrict these parts. We are gonna purify, put together in a tube, and perform an assembly reaction. In fact, for all this step, or to optimize all this step, this cyanobacter system has developed an assembly portal that we can choose the different parts that we want to include in our final vector, as cyanobacterial replicants, antibiotic markers, promoters, E. coli origin of replication, etc. Another part that we can use in our constructions are promoters. Promoter is a region located near the transcription start sites and regulate transcription initiation of the gene by controlling the binding of RNA polymerase. In this table, you can see the most common promoters that has been used in cyanobacteria. We include some constitutive promoters that are which expression is constant along the time. In this list, we have the PCP 560 that is a super strong promoter from Cyanocystis, and PSBA2 from Cyanocystis that is a promoter from one of the subunits of the PS2. We have also some indicible promoters which respond to external input, normally to the concentration of our inducer. And in this list, we have this one. We have some specific one that has been developed for E. coli, but they work in cyanobacteria, PTRC, PLAC. Recently developed for Cyanocystis, L03 that is indicible by anhydrous tetracycline. And also one endogenous promoter from Cyanocystis, PNRSB, that is indicible by nickel. This is a small list of the promoters that have been used in cyanobacteria, but more and more has been discovering more promoters. In this list, we have some new indicible promoters that has been described for cyanobacteria. I'm gonna explain the last 3 ones. We have in the first place promoter indicible by vanillate. In absence of vanillate, the RNA polymerase is not going to be able to join to the operator of this promoter because the repressor is going to be dimerized and join to this operator. In presence of vanillate, this molecule is going to join to the repressor, liberating the space for in the operator in the promoter, allowing to the RNA polymerase to join and transcribe the gene of interest. In the second place, we have a PVAC promoter that has been studied many years in E. coli. In absence of arabinoids, that is the inducer of this promoter, the repressor RC is going to join to the one of the regions of the promoter and to the operator, forming a secondary structure in the promoter and not allowing to the RNA polymerase to join. When arabinose is present, the is going to join to the repressor RC that is going to change or release this secondary structure because some part joins to the other region of the promoter, I2, and allowing to the RNA polymerase to join to the promoter and transcribe the gene of interest. And in the last place, we have a rannose indicible promoter. In absence of rannose, the activator RAS is not going to be able to join to the regions of the promoter and dimerize. But when we add rannose, this activator dimerize joins to the promoter and allows the RNA polymerase to join and also transcribe the gene of interest. Next toolbox that we can use in cyanobacteria to modulate the expression of the gene of interest could be ribosome binding sites. Ribosome binding sites are sequence upstream of the start codon that is responsible for the recruitment of the ribosome during the initiation of protein translation. This ribosome binding site normally contains the Cyan-Dalgarno sequence. In cyanobacteria, different types of ribosome binding sites have been used. We can use native ribosome binding site, synthetic, or heterologous one. The effectiveness of riboswitch is going to depend on the base pairing potential with the anti-Cyan-Dalgarno sequence and the spacing between the ribosome binding sites and the start codon. In this experiment, they have used 3 ribosome binding sites from the BioBrick system, and also they have developed a ribosome binding site synthetic one for Cyanocystis. And they compare the level of expression of the fluorescent protein with these ribosome binding sites in Cyanocystis and in E. coli. As you can see here, the one that works better in Cyanocystis was the synthetic one. And the important message from this slide and from this graph is that we can say that it's really, really important to characterize these biological parts in the respective cellular contents, because the things that can work from E. coli, they cannot work for cyanobacteria and vice versa. In the second toolbox that we are going to describe in this presentation are riboswitches that are short sequences or messenger RNA that can change their structural conformation to regulate gene expression. The most famous riboswitch is used in cyanobacteria is a theophylline dependent riboswitch. In absence of this theophylline, the ribosome binding site is going to form an aptamer with the part of the sequence of the messenger RNA, not allowing to the ribosome to join to this RNA and not translating the gene of interest. But when we have theophylline in our system, the theophylline is going to join to this aptamer. The ribosome binding site is going to be liberated, allowing to the ribosome to join and translate the gene of interest. This theophylline dependent riboswitch has been tested with different promoters. In the first place, they has been tested with PTRC, a very strong promoter, but it's very leaky and absent of inducer. Using this riboswitch, this promoter can be less leaky in absence of the inducer, in both inducer, and also can increase the induction ratio after adding both inducer. Also, this riboswitch has been used with some constitutive promoters as PCON2. This is an example for Synechococcus, and they tested different riboswitches, modifications of these theophylline-dependent riboswitches, some changes in nucleotides. And as you can see here, the induction ratio, these different versions, is pretty good. But the important part is having a system that is off when we don't have the theophylline present. So you can see here in these two cases, in absence of theophylline, we have still expressing. The next toolbox that we can use in our system are terminators that are DNA sequence that signal the RNA polymerase to terminate transcription and improves the stability of gene transcript during translation. In this example, They have used a battery of different terminators for E. coli, synthetics, or different fates. They have been using a system where they use the Ranose promoter and a yellow fluorescent protein, and they have introduced the different terminators between the promoter and the fluorescent protein. In the case of they are going to have a control with our terminator that is going to produce the higher levels of fluorescence, as you can see here. And the idea is to test which terminators are able to reduce the amount of fluorescence that we are going to get after inducing the promoter. And as you can see here, most of them, the E. coli terminators and synthetic terminators work very well, not the phase 1. One of the main problems when we want to introduce modification in the genome of cyanobacteria is the amount of antibiotic resistance that we can use. Because more modification, more antibiotic resistance that we need to introduce in the chromosome. That also there could be a burden for the cell. Or to increase the number of modifications that we can perform. Some markerless system have been developed for cyanobacteria. In this first one, it's a very old one, but I have been using in my thesis and it works very, very well. The main problem is we need to build 2 plasmids for its modification. We are going to start with a wild type strain, and we are going to modify the wild type RPS12 gene. We're going to introduce a mutation in the nucleotide 128, change from adenine to a guanidine. And doing this modification, our strain is going to be, our gene is going to be mutated, and our strain is going to be streptomycin resistant. After that, we are going to perform a transformation using a plasmid that is going to target our gene by introducing RPS12 copy of from Cyanocystis and their RPSBA1 promoter and actinomycin resistant. This construction is going to recombine with the chromosome, and this part is going to be integrating in the chromosome. And we are going to be able to select our cells because they are going to be streptomycin sensitive, because the RPS12 wild type copy or Cyanocystis is going to be dominant against the mutated copy. And also we are going to be able to select because our cells are going to be kanamycin resistant since we have introduced this cassette. In the second step, we are going to use a second plasmid that contains either a target gene mutated or absorbed version of a gene if we want to delete it. And it's going to recombine with the previous gene modified. And in this case, our cells are going to be streptomycin resistant because the wild type copy of RPS12 is going to be removed, and kanamycin sensitive because we don't have the kanamycin resistant anymore. A new system that has been developed recently for cyanobacteria is CRISPR-Cas technology. This CRISPR-Cas system, we need only one plasmid that contains a kanamycin resistant gene. And also we have our Cas protein and the indicible promoter, PILAC, and the CRISPR RNA coding sequence and their constitutive promoter. And we need to include also in this plasmid upstream another downstream sequence of the area that we want to modify. When we have built this plasmid, we introduce in cyanobacteria and we activate the expression of Cas by using IPTG. The system is going to express the Cas protein and the CRISPR RNA, and it's going to target the genome sequence that we want to modify. And the Cas protein is going to cleavage the DNA, and only the cells that are going to produce homologous recombination with the regions that we introduce in the plasmid are going to be able to survive. And in this system, we are going to select our cells because they are going to be kanamycin resistant for the cassette that we introduced. And to get rid of the plasmid, we are going to need to segregate the constructions by growing the cells in VG11 without kanamycin. In this last part of the presentation, I'm going to show some examples how to metabolic engineering has been developed in cyanobacteria. In this diagram, in this article, they have summarized which compounds has been produced by metabolic engineering in cyanobacteria. As you can see, there is a long list: isobutanol, ethanol, mannitol, sucrose. In this presentation, I'm going to explain two examples. In the first one, fatty acid biosynthesis, and in the second one, limonene production. Fatty acid biosynthesis in cyanobacteria is characterized for three steps. In the first step, the initiation step, acetyl coenzyme A is transformed to acetoacetyl ACP for three enzymes, acetyl-CoA carboxylase, fat D, and fat H. This molecule is after that used in the elongation cycle that is performed several times until the fatty acid contains 16 or 18 carbon. These fatty acids are used in the saturation step. Depending on the cyanobacterial species, we are going to have different amount of the saturases. All of them contain at least the C that is going to introduce the first double bond. But for example, 7942 doesn't contain any more. 7002 contains two extra one, this A and this B, that in 7942 only contains the C. And in order to do that, they use this A and this B from 7002. And the first information that we can start from this publication is the production of the desired compound is going to depend on the promoter strength. These authors, they use two different promoters to express these desaturases. PNRSB, as I mentioned before, is indicible by nickel, and PTRC that is indicible by IPTG. As you can see here, with PNRSB, the level of alpha-linolenic produced was very low, and these levels were higher when PTRC promoter was used. These levels were quite similar to the origin species where these desaturases were extracted, 7002. In fact, in this article, they analyze the activity of these two promoters using Lux AB construction. They measure the luminescence along the time after induction with the different inducer. And as you can see here, the maximum level of expression for PNRSB, it takes place at 24 hours, is around 3,000 relative luminescence units. And also they measure PTRC with different concentration of IPTG, observing that there is no difference between 100 micromolar and 1 millimolar IPTG, and getting the highest levels of expression in 24 hours, and there are around 25,000 relative luminescence units, indicated that with this experiment that PTRC is stronger than PNRSB, and that's why we get more alpha-linolenic with PTRC. In order to increase the omega, the specific production of alpha-linolenic, we could think that we should have more intermediate to increase the production of the final product. In order to do that, in this article, they overexpress different enzymes involved in the elongation step, and they observe that only with fat F, they decrease all the C14 and C16 fatty acid species, and they increase C18 species that are interesting for the production of alpha-linolenic. They got a similar result when they deleted fat D, that is a gene involved in the degradation of fatty acids. As you can see here, with this modification, they decrease C16-1 and increase the C18 species. Combining these two modifications in the strain that contains the desaturases, they got a high increase in the alpha-linolenic. In this case, was 22.5% of alpha-linolenic in the cells, compared with only PTRC test AB that was around 8%. In the next sample, I'm going to talk about limonene production. I'm going to starts explaining why limonene is a very interesting molecule. It's a molecule that can be used as a fragrance, a flavor in different drinks. Applied to biofuel production. In cyanobacteria, we can produce limonene, only introduce the limonene synthase, and using the precursors obtained in the methyl erythritol limonene production, but we can have some negative effects in other metabolic pathways. In this case, the production of limonene has a negative impact, as you can see here in the proteomics analysis and here in the phycobiliproteins content, no changes in chlorophyll, in carotenoid, but it was observed a decrease in the phycobilin content. And also another negative impact of limonene production was a decrease in the levels of, or in the oxygen evolution, in the photosynthetic activity. This was mainly because the synthesis of limonene doesn't require higher contents or NADPH ATP ratio, and that's why the cells, they don't need to they don't need to use too much light to use these electrons to produce ATP and NADPH. And also, when we want to produce a specific compound, normally we can choose a specific enzyme from an organism, but using or testing different, the same protein from different organisms, we can get different catalytic activity, and it can influence the yield of the desired compound. In this experiment, they measure the limonene production using 2 limonene synthase, in the first case from Menta spicata, and in the second place from citrus lemon. And with the Menta spicata, the levels of limonene were higher at different times post-induction. And sometimes we can modify without only identify some competitive pathways to improve the production of specific compound, but also we can use the genome scale models that I explained previously in this presentation, using algorithms like ODFORS. In this case, they use a model from Negothistis, and they optimize in the model the production of limonene. And in the first place, they identified that the overexpression of ribulose 5-phosphate epimerase and isomerase was enough to increase around 89% of the production of limonene in silico. Combining this overexpression with the overexpression of the geranyl diphosphate synthase, the production increases 92%. Once they identified this modification in silico, the authors decided to test these modifications in vivo. And as you can see here, they always compare with the original strains, only expressing limonene synthase. When they are RPAI and RPAE, the limonene production increases. The same when they are only GPPS, and when they combine all the modification in the same strain, the levels increasing very highly. And also, as I mentioned before, when we express a non-native pathways, we can divert resources from competitive pathways to design, to generate the specific compound. And in this case, they didn't observe any changes in carotenoids, but when they combine these modifications in the strain, they observe a decrease in the chlorophyll content. In fact, the pathway to produce chlorophyll is competing for the synthesis of limonene. And I would like to finish this presentation showing some future challenges in metabolic engineering of cyanobacteria. We can develop better synthetic biology tools in order to perform metabolic engineering. We can increase the speed and efficiency of genome engineering, maybe in the future reducing polyploidy. In order to do that, we need to figure out why cyanobacteria has multiple copies of the chromosome inside the cells. And as I mentioned before, we have in cyanobacteria, they have been developed CRISPR-Cas system, but the efficiency of this sometimes this CRISPR-Cas is able to target sequence that we don't expect. And improving this technology that only requires one plasmid instead of the all marker-less system that requires two, we are going to speed up the ability of getting modifications in cyanobacteria. And also, instead of using integrated chromosomes, that is for metabolic engineering is most of the cases, if we are able to perform new replicative plasmids to express, for instance, DNA as they have been used in E. coli and yeast, we can develop or libraries that can be analyzed by high throughput screening, increasing the ability and the speed of what we can optimize the production of a specific compound. And also, we need to develop better regulatory control to increase strain stability, because as I mentioned before, most of the tools that we have used from in cyanobacteria, they have been developed for E. coli and other prokaryotic organisms. And if we have more specific tools for cyanobacteria, probably we are going to be able to control better the expression of our genes. And also, this is one idea that has been recently developed for E. coli, is the automation of iterative design, build, test, learn cycles. It has been used in E. coli to produce flavonoids or dodecanol. And in this case, instead of doing all these steps in the laboratory, most of the steps are performed by computers. In the process of design, we are going to use the in silico models to identify the pathway that we want to express. For example, computers are going to design the parts, experiments, instructions. We are going to, instead of doing PCR in the laboratory, we are going to synthesize the DNAs. We are going to put all the parts together using robots. We are going to test our battery of libraries using high throughput screening. And with all this data, we can using machine learning and all computer tools to analyze the data and start a new cycle or design, build, test, and learn. And the last slide of this presentation, I'm going to show some useful resources, some new or recently reviews related with metabolic engineering cyanobacteria, and also some tools or databases that can be useful to build and to identify the pathways that you want to use. And thank you.